Hello, good evening.
Good evening, sir.
So last class we discussed about the Koch postulates, drawbacks, molecular Koch postulate, pure culture preparation. For that we need solidifying agent. The beta solidifying agent is agar over the gelatin, and there are several advantages of agar we have discussed. Today's class is establishment of other two different fields, medical microbiology and immunology. Establishment of the fields of medical microbiology and immunology. First of all, you should know the meaning of medical microbiology and immunology. These two fields are well connected. These two fields are well connected because in medical microbiology, this subject is deal with various infected diseases in human or other animal, and their disease symptoms, as well as how to treat such pathogenic diseases. So this overall knowledge given by the subject, that is medical microbiology, and is a connected part. Another field is called immunology. Immunology is basically about to gather the knowledge of our immune system. Immune system, what is immunity? That means prevention or blocking the activity of pathogenic organism that affect also present in our body itself, and that property is called immunity, and the subject is known as immunology. So immunology, this subject deals with the concept of immunity. Concept of immunity which is actually present in our body and continuously fighting against the pathogenic organism, disease causing organism, and try to eliminate or kill them. And there are different types of immunity we are discussing. Other semester, in some semester, has the concept of immunology. Okay, so how these two fields are come in our regulated course because of contribution of some scientists. Some scientists' contribution make this happen. These two fields, medical microbiology and immunology, as a subject, as a regulatory subject, as a field, and two important fields. So these two fields are established by some notable contribution of some scientists. And we are starting with the important contribution of well-known scientist Edward Jenner. Edward Jenner, he immunized the people against smallpox. Immunized the people against smallpox. One of the deadliest disease, high mortality rate containing disease, smallpox. That means out of 100, more than 90 people after die if they are infected with smallpox. Smallpox actually caused by virus, smallpox virus, right? So, huh? 90% mortality, 90% and above. Almost every single case they are died. Just 5 to 10 people maybe survive, but maximum time they are died if smallpox is infected. So one of the deadliest disease at that time. But nowadays, smallpox is totally eradicated. Totally eradicated by the use of vaccine. That is another concept we are coming to that. Okay, so Edward Jenner immunized the people by one of the deadliest diseases known smallpox. How? For that, he used he introduced materials from cowpox, which protects them, which protects the people from smallpox. So how he immunized the people? He just used the another disease that caused in the cow, not in the human. The disease known as cowpox, and it is also a virus disease, but different virus. Okay, so just he introduced the cowpox materials. That means he collected the fluid from the cowpox lesion and introduced into the patient that suffered from the smallpox. And miraculously, the patients, that means that they who was suffered from the smallpox, they are actually cured. Clear? From that, the vaccine concept is collected or came into the fact. The vaccine concept, the word vaccine do not give by the Jenner, it is actually given by the Pasteur in the next scientist contribution. There is a my contribution list number two, Louis Pasteur. Louis Pasteur has enough more than Jenner's contribution. That means enough role in the medical microbiology and immunology. Louis Pasteur's contribution was there in the spontaneous generation disproving. Also in the field of establishment of medical microbiology and immunology. So that's the idea of Jenner. He introduced cowpox material and introduced into the smallpox patient. And smallpox actually cured by the happen by that. Jenner's idea is clear, but the question was how the cowpox material actually cured the smallpox. My question is how cowpox material works against smallpox. Against smallpox. That is the main concept, main question after the Jenner discovery. How it works, how the cowpox material works, and it cured the smallpox virus. It eliminates the smallpox virus.
Sir, the cowpox material acts as an antigen. So when it is
Okay.
introduced in the body, the body will, the B lymphocyte cells will produce antibodies against it. And when the actual disease, the smallpox disease will occur, so already antibodies are ready for it to fight against it. And it neutralizes the effect by binding with it.
Okay. That's correct. Janvi said that cowpox material actually acts as an antigen, and when our immune system recognize that antigen, it produce antibody. But cowpox antibody can work on smallpox. That is the ultimate question. Cowpox is different virus and the smallpox is different virus. Can this material, that means the production material from the cowpox, works against the smallpox? My question is clear, Janvi. My question is clear. Can
Yes,
cowpox material production of antibody? It is produced antibody, clear, but that antibody can react with smallpox also, because smallpox is different virus. Can works
so
against the smallpox.
that I don't know.
Okay. For that answer, we are looking for the answer at the end of the topic discussion. Before that, we are discussing another some other scientist contribution. Okay. We are uncovered this question answer at the end, right? The second scientist contribution is none other than Louis Pasteur. Louis Pasteur. Because this answer need this concept actually related to a complex immunology. It works. Okay, we are discuss at the end because it needs the concept of immunology more to answer this. So Louis Pasteur contribution after the Jenner discovery, Louis Pasteur discovered that Louis Pasteur and his colleague named Rox discovered that an important thing, that is, when a bacteria or other microbes, basically you work in bacteria, incubated in cultures for a long time, then they lost their disease causing ability. So Louis Pasteur and his colleague Rox discovered is another new thing. What is the concept? The concept is when you grow the bacteria in the laboratory culture for many times, for many years or months, from the body, outside from the host, they culture in the laboratory condition. Okay, outside from the human host or outside from any animal host, and they watch that after incubation for the long time, the disease causing ability, that means the virulence property of that bacteria become lost. That means after so many time, when you inject the material into the healthy host, the host will survive easily and no disease symptoms are there. Clear? So that is the important concept they discovered, Rouse and Louis Pasteur, and he discovered from the bacteria. And he called them bacteria, that disease causing ability lost bacteria, attenuated or weak bacteria. Pasteur called weak or attenuated because they are not dead. They are not, these bacteria are not dead. They are live, they are growing, but just the difference is that they lose the capacity of disease formation. Clear? Lose the virulency property. But they are easily grown, they are multiplied, they are metabolized, everything they have done, but just they lose the disease causing ability. And that's why they termed as this group of microbes weak strain or attenuated strain. And nowadays we know that these weak strains are actually used for the vaccine preparation. Yes or no? Attenuated strain, weak strain are used for the vaccine preparation.
Yes.
And from that observation of the Louis Pasteur and Roux, various vaccines are developed. Pasteur actually developed vaccine, first vaccine, actual vaccine, and the term vaccine. Next point. From that concept, Pasteur discovered rabies vaccine. Another, another deadly microbes, deadly disease, rabies. 100% mortality rate. Smallpox 90%, over 90%, it is 100%. That means every person will die if they infected with the rabies. And still also now, some people are died because of lack of knowledge. When any animal scratch, not only bite, scratch also cause infected rabies virus to infect. Scratching, biting, so for sure you need to do vaccination. But for the scratching also, you need vaccination and go for the vaccination within 24 hours. It is 100% necessary. Please educate and try to educate other because still in rabies case found in the India because of lack of knowledge and negligence. We try to negligence that it's, it's just scratch. What will happen? It happens. It happens after a month or after 2 months or 3 months. And after if the symptom is come, nobody will survive. Clear? So that's the deadliest about the rabies vaccine, rabies virus, and the rabies disease. So that disease vaccines are created by the Louis Pasteur. Clear?
Yes.
How he did? How he did that? How rabies vaccine he observed? He incubated a rabbit with saliva from rabbit dog. See the step number one. Rabbit dog actually contain virus in the saliva region, mouth parts. Okay, So he just collected the saliva from the rabbit dog and introduced into the rabbit, a healthy rabbit. Rabbit is the here model organism for the preparation. Then he extracted the virus from rabbit spinal cord. After isolation, he attenuated the strain. How does he attenuate it? That means he grow inside the laboratory condition for multiple time. That means it could be month or year. And ultimately the concept will follow. This concept, if you grow the bacteria or virus or any microbes outside from the body, outside from the host and the laboratory condition you grow for the several period, ultimately the disease causing ability will go. And that's concept he applied. After the extraction, he attenuated the strain, attenuated the virus with the virus, and inject into the patient. And patient will survive. So that's the concept of this concept actually is the vaccination concept. Vaccine preparation concept and the process is called vaccination. Inject into the patient.
Sir, I have a question. Sir,
Hmm.
when the virus has lost its disease-causing ability, when it's
Right.
cultured for a long time, then
Ah,
after that it is inoculated into the patient. So,
yes.
how can antibodies be formed? Because it has already lost its disease-causing ability.
Okay. Antibodies actually form from the outer structure of the surface. Janvik has questioned. He questioned that if the disease causing ability is lost, how does the antibodies will produce in our body? Because antibody is the molecule, the protein molecule that produce against the foreign antigen just outside the structure. They observe antibodies actually observe or recognize the outer structure of the antigen, not any in detail structure, in depth structure, inside structure or any kind, anything. That means if the disease causing ability will lost, the antibody is still produced because the structure is the intact. The structure of the microbes is intact.
Okay, sir.
Clear? So that's why the antibody is still produced, and they try to now eliminate the looks like same microbes or looks like same virus when it enter clearly. So that's the concept of vaccination. You introduce the live vaccine, live pathogen, but the pathogen should be weakened. The pathogen should be attenuated, or for the safer case, nowadays we are given killed vaccine, that means killed microbes, safer purpose, but it has some disadvantage. Some advantage, disadvantage always there. Okay, so just introduce live bacteria as a weakened form or killed bacteria, right? If the bacteria is intact, our body system recognize that, and after recognition, they produce antibodies and other molecules that will kill or neutralize them. That is the overall scenario of vaccination. Just like a COVID vaccine. In 2020, we are all are giving the COVID vaccine. Now, what is the concept of COVID vaccine? Giving the COVID vaccine because COVID vaccine, if the COVID, the virus will come in our body. In previous system, we has already antibody. That means kill bacteria, kill virus already given, and the antibody always resin in our body and try to eliminate it further thing. That is the concept. That is the concept of vaccine preparation. and the process is known as vaccination. Is ultimately achieved by this concept. From this concept, Louis Pasteur. And Louis Pasteur actually coined the term vaccination. Edward Jenner prepared that, but as a unknowingly he done. He do not know even that it is actually cured. It will happen or not. That means the ultimate goal to the outcome he do not know. In Edward Jenner case, just a given. Which is the first main vaccine launched? Louis Pasteur. Rabies. Smallpox. Yes, he honored Louis Pasteur honored Edward Jenner. So actually Edward Jenner first. But if you conceptualize that, rabies vaccine is the first because Louis Pasteur actually works like a vaccine that we are nowadays we are using. That concept we are now using nowadays also in modern concept. Clear? So vaccination theory clear. So from that, Louis Pasteur,
Yes.
Louis Pasteur used the word, coined the term vaccination in honor Edward Jenner. In honor to Edward Jenner, because Edward Jenner material was cowpox, cow materials, and the vaccination is the Latin word came from the word vacca, and that meaning is cow. That means cow. Vacca word actual means cow. So in honor to Edward Jenner, material that he produced from the cow lesion, he word the term vaccine, vaccination. Clear? That concept clear? So Louis Pasteur utilized this concept to make another vaccine called anthrax vaccine. Pasteur also prepared anthrax vaccine. Anthrax as a disease established by which scientist? Anthrax as a disease established by which scientist? Is it Louis Pasteur or other? Answer. Louis Pasteur discovered the vaccine against the anthrax, but as a disease, who established that it caused microbes? Robert Koch. Robert Koch in his Koch postulate. In his Koch postulate, what is the Koch postulate? Overall things it came from actually to anthrax disease. The disease anthrax, he gave that 4 laws or 4 steps to identify a disease causing organism. So it is was Robert Koch who discovered the anthrax as a disease that cause in effect of animals, not in human. Some form also happens in the human, also cause disease symptoms in the human, but mainly it cause in the animals. Okay, so Robert Koch discovered the anthrax as a disease, but Louis Pasteur discovered the vaccine against the anthrax. Clear? So anthrax vaccine discovered by Louis Pasteur. And also Pasteur discovered in next cholera vaccine. Clear? তো লোয়ার ক্লাসে আমরা কি পড়ে আসতাম ম্যাক্সিমাম কনসেপ্ট ভ্যাকসিনেশন মিন্স এডওয়ার্ড জেনার বুঝতাম, হ্যাঁ কি না? বাট অ্যাকচুয়াল এডওয়ার্ড জেনার তো করেইছে, ঠিক আছে, এডওয়ার্ড জেনার তো মিস্টেক ওটাকে ইউজ করেছে ফর দ্য পারপাস, হি নোজ নাথিং অ্যাবাউট ইট, বাট actual concept of vaccine and various vaccine preparation is came from the what? Louis Pasteur. Several vaccines are given: rabies vaccine, cholera vaccine, anthrax vaccine, and prepared by the same method. That is the main concept. Grow outside into the laboratory for several period, and they lost their ability to cause disease. They are weakened stem, and these weakened stem can be used for the making of vaccine. Louis Pasteur contribution clear from the medical microbiology aspect. Everyone,
Yes, sir.
scientist number 3 in my list, von Behring and Kitasato. Von Behring and Kitasato, Japanese scientist. Kitasato. They demonstrate that white blood cell, that is WBCs, another name of WBC is leukocyte, white blood cells, and they are produced chemical. Play in immunity. So one of the blood component he recognized, and the blood component blood cell is WBC, and they have immunity role in immune system development. Not only their role, they actually produced chemicals. What type of chemicals? Protein molecule, soluble protein molecule. These chemicals are soluble protein molecule observed by Kitasato and Behring. These produced chemicals are actually soluble protein molecule, and they call that protein molecules as antibodies. যে আমরা এতক্ষণ ধরে অ্যান্টিবডি অ্যান্টিবডি করে চেঁচাচ্ছি, body produce antibody. Previous day we are not knowing about any kind of things that antibodies. After the discovery of Behring and Kitasato, they actually demonstrate that how does our immune system work. It does not work by the every cell in the blood, mainly worked by WBCs, white blood cells in our blood, right? And the WBC actually producing that type of chemicals, that is called antibodies, which is the Y-shaped structure and 4 subunits are present, 2 heavy chain, 2 light chain containing Y-shaped structure. So not only they just demonstrate the production material, they actually given the structure of antibodies also. Clear? Behring and Kitasato concept, they discovered the soluble protein molecule which is synthesized by the WBC, and that is antibodies. From that concept, Behring and Kitasato discovered another new thing. Next point, from their observation, discovery point of view, they discovered antitoxin against diphtheria. Diphtheria toxin. Diphtheria actually a disease, okay, bacterial disease, bacterial causative agent caused by the bacteria, and the scientific name of bacteria Corynebacterium diphtheria. Corynebacterium diphtheria produce toxin material which is known as diphtheria toxin. And this diphtheria toxin has the activity of virulence activity. That means produce symptoms and cause disease. Virulence activity. So not by the individual bacteria, bacteria secrete some protein which is known as toxin. That toxin is called diphtheria toxin, and diphtheria toxin cause the symptoms, cause the disease symptoms. That means here the virulence power is the toxin, not the whole bacteria. Clear?
Yes, sir.
I repeated, I stressed that the line that Corynebacteria diphtheria release toxin cause the virulence, not the individual bacteria. Bacteria do not think anything outside structure. So the vaccine concept, which is given by the Louis Pasteur, is it work in this scenario? Louis Pasteur vaccine concept is working against this scenario? Huh? Yes or no? Bhalo, think twice. Here full ব্যাকটেরিয়া কিছুই করছে না এখানে, এখানে ব্যাকটেরিয়া রিলিজ টক্সিন করছে। তো তুমি যদি ব্যাকটেরিয়াকে অ্যাটেনুয়েটেড ফর্মে ঢোকাও কি হবে? Just think one time, if bacteria is introduced into the attenuated form or weakened form, that is the concept of Louis Pasteur. Is it curable, or is it cured against the diphtheria? হবে না তো কিছু, কারণ তোমার তো একটা ব্যাকটেরিয়া কিছুই করছে না, এখানে কি করছে? ব্যাকটেরিয়া প্রেজেন্স আছে and they are releasing toxins, and these toxins actually create the symptoms. Yes or no? So তোমাকে কি করতে হবে? What you have to do? You start to produce against the toxin antibody, antitoxin antibody, which is known as, actually simplified, antitoxin. So here against the bacteria, antibody against the bacteria could not do anything. না ভায়োলেশন না, ওর কনসেপ্টটা অন্য ছিল। ওর যে ব্যাকটেরিয়া গুলো বা ভাইরাস গুলো কাজ করলো ইন্ডিভিজুয়াল লেভেলে কাজ করছিল, কোন রকম টক্সিন রিলিজ সিক্রেট করে করছিল না। তো এটা ভায়োলেশন না, বাট এটা আনাদার মেকানিজম, আনাদার থিং, নট ভায়োলেশন। কারণ ভায়োলেশন মানে কি, ওটাকে তোমাকে ডিসপ্রুভ করতে হবে, ভায়োলেশন তো হচ্ছে না, বাট এটা আনাদার থিওরি। So some bacteria individually do not do anything, but they produce or secrete toxin, and that toxin create the damage in our body. Clear? So আমাকে কি করতে হবে তখন তখন এদের কনসেপ্টটা লাগছে মানে কিতাসাতো and Behring concept. Antibodies against the toxin, produce antibodies against the toxin, not for the bacteria that create the what, that reduce the symptoms. Yes or no? So from that concept, the antitoxin produce. So what is antitoxin and how they produce? A simplified chart. They isolate diphtheria toxin first. Diphtheria toxin, not bacteria. See, isolate diphtheria toxin. Clear? Inactivate them. Inactivate toxin by heat or chemicals. Inactivate the toxin, not the bacteria. Clear? Inactivate the toxin and then introduce into the rabbit. And the rabbit produce antibody against diphtheria toxin, and this antibody particularly specifically bind and neutralize. So if you inject this type of antitoxin in the patient, the symptom slowly disappear. Yes or no? বাট আমি যদি ব্যাকটেরিয়ার এগেইনস্টে ভ্যাকসিন প্রিপেয়ার করে ইনজেক্ট করতাম পেশেন্টের, কিছু লাভ হতো? সিম্পটম রিডিউস হতো? পসিবল না, because not the bacteria is caused by a toxin. So that's why Kitasato and Behring do toxin isolation, not bacteria. Inactivate them, inject into the rabbit, produce antibodies, that means antitoxin, and inject into the patient, and patient slowly recover. So after the antibody, which molecule actually works to protect our cell? So that is the antibody discovery, the antitoxin produced. Clear? Everyone clear? Behring Kitasato contribution in medical microbiology field. Those are online.
Yes, sir.
Okay. Science number 4. Elie Metchnikoff. He discovered the process of phagocytosis. Process of phagocytosis. What is phagocytosis? Do you get the term before phagocytosis in class 11, 12 or lower class? Define, try to define phagocytosis process. Monomer try করবে phagocytosis. Anyone from the online? Phagocytosis.
Sir, a white blood cell destroys, like the way it destroys a foreign particle, is it called phagocytosis?
Destroy the foreign particle, correct, by antibody production or some other process. Some white blood cells also produce antibody. In the previous concept, we know that WBCs produce antibody that kill or neutralize the pathogen, and pathogen producing any virulence agent like toxin. That the phagocytosis concept is same or they are different?
Sir, the phagocytes surround the cell and then kill it.
Okay, that means totally ingestion, eaten up, খেয়ে নেওয়া, clear.
Sir, I can't hear your voice.
Yes, sir, your voice is.
Now audio clear?
Yes, sir.
Clear? Okay. So phagocytosis means eaten up the pathogen, eaten up the microbes. Just
Sir, the cell engulfs the pathogen and destroys it.
the WBC itself, WBC creating this phagocytosis process. But the cell is different. WBC has different variety, neutrophil, phagocyte, monocyte, etc. Different WBCs are there. Some of the WBC cause phagocytic activity. That means, what is phagocytosis? Phagocytosis means engulfing. A disease causing bacteria. And those cells involved in the phagocytosis process called phagocytes. Called phagocytes. Some of the WBC, white blood cell, has the ability to phagocytosis, to involve in phagocytosis process. Some example, monocyte neutrophil, part of the WBC, yes or no? Monocyte neutrophils, they are the part of the WBCs family, but they do not produce any kinds of antibodies. Instead, they are just engulf the materials, engulf the foreign materials like pathogen, and by that way they kill the pathogen. So two-way WBC can work. One is the production of antibody. By the production of antibody, it can kill or neutralize the pathogen, and that concept given by the Kitasato and the von Behring. Okay, and another mechanism to killing the pathogen in our immune system is phagocytic process, and that concept given by Elie Metchnikoff. Both the scientists are winner of Nobel? Discovery. Clear? In a medical field, in a medical একটাই biology field একটাই থাকে, physiology, medicine and physiology. So overall immune system, immunology as a concept, overall just a knowledge, clear or not? How does our immune system work against the pathogen? It can be two-way attack, antibody production or phagocytosis. Some concepts are clear. So by utilizing the concept, now try to answer how Jenner's cowpox material work against the smallpox. How Jenner's cowpox material works against the smallpox. Smallpox কি কোনো smallpox ব্যাকটেরিয়া দিয়ে কি ভাইরাস এই কি সংক্রমণ করে নাকি ওর কোনো মানে এরকম টক্সিন দিয়ে কিছু? না টক্সিন না, ভাইরাস, টোটাল ভাইরাস। প্রবলেম ক্রিয়েটার হচ্ছে মানে ভিরুলেন্স এফেক্ট টোটাল ভাইরাস। Any guess? না না, অ্যান্টিবডি প্রোডাকশন, এখানে ফ্যাগোসাইটোসিস হবে না, অ্যান্টিবডি প্রোডাকশন। অ্যান্টিবডি কনসেপ্ট আমরা পড়লাম, তো বাই দ্যাট নলেজ anything, any concept of how cowpox materials are used or works? Physiological condition is same, common as like cowpox, smallpox. On the track, কাছে আছো, আরেকটু এগো। See, cowpox and smallpox. Cowpox caused by the cowpox virus, vaccinia virus actually, and smallpox caused by the smallpox virus. Virus could be different, but their overall structure, আমরা কি just discuss করলাম কি যে structure is actually determining the what types of antibody produced in our body. Overall structure, majority of the structure is homologous, that means similar. Cowpox virus structure and the smallpox virus structure, though they works against the different infected different animals, one is infect a cow and other is human, but their structure, overall simplicity of structure is same. So now just thought, if the cowpox materials is given to the patient, cowpox cannot infect into the human, but the virus structure is there, and that recognize our body cell WBCs and antibody produced against them. Yes or no? Yes. And that antibody can work if smallpox virus is present in body system. Clear? জেনার কিছুই জানতো না এই সমস্ত ব্যাপার, জাস্ট জেনার ট্রায়াল করেছে একজন, ট্রায়াল হ্যাঁ ট্রায়াল, বাট তুমি যদি আজকে জেনারেল জায়গায় দাঁড়াও এই কেস করতে পারবে না, একটা হিউম্যানের এগেইনস্টে তুমি যে কোনো কিছু ম্যাটেরিয়াল ঢুকিয়ে দিতে পারবে না, বাট তখন কিচ্ছু করার ছিল না, 17th সেঞ্চুরি টাইম। জাস্ট জাস্ট ট্রাই, জাস্ট ট্রাই, হ্যাঁ, যদি মিরাকল হয়ে যায়, and actually it caused miracle. But the overall concept is this: the cowpox virus structure, which is much more similar to the smallpox virus, and when they injected the cowpox material, it do not cause any infection in the human body, but the structure similarity make the production of antibody, that antibody kill or neutralize the smallpox. And that's the concept, the beautiful concept, complicated but yet beautiful concept of immunology called cross immunization. এই কনসেপ্টটা কি বলে? That concept is known as cross immunization. That means one antibody production produced against the one antigen that can affect also second antigen. Antigen 1 antibody produced against the antigen 1 can also affect the second antigen, provided the structure will be more or less similar, maximum similarity. Clear? Cross immunization. So Jenner's vaccination, totally concept based on cross immunization. না, just trial, just trial, just last try. But Pasteur know that how bacteria can be weakened, how bacteria can produce, the weakened bacteria can work against the pathogen. They know. Pasteur know.
সেক্ষেত্রে আমাদের কাছে এতটা কিছু আছে, এটাই কারণ যে কেবলমাত্র মিউটেশনটাই কি আমাদের প্রবলেম করে। যেমন ভ্যাকসিন তৈরি করার না, আমাদের কাছে গেস রয়েছে, কোভিড ভ্যাকসিন তৈরি করবে একটা কারণে মিউটেশন রেগুলার মেইনটেন্সে
changes. See, same thing. If mutation occur regular base, the structure, outer structure could be changed, and that cannot be recognized by the same things, cannot be recognized by the body, and that create problem in vaccine generation, and that is why maximum time vaccines are failed because of mutation issue. The RNA virus, basically RNA viruses has the tendency to do havoc mutation in your body, in the structure of the virus body, and they change the overall structure of the virus particle, and same time they could not recognize by the body that time. Clear? So problem in basically the RNA virus, maximum time, and COVID was RNA virus. ওটা তো ডিটেকশন প্রসেস, but actually RNA virus. That's why different strains are come within the regular interval, delta, epsilon, etc. Okay. So concept of medical microbiology immunology এখনো বাকি আছে। So Elie Metchnikoff, clear? Online students, did you hear
Yes,
my
sir.
voice?
Yes, sir.
Number 5. So after the overall knowledge generated by in immunity, scientists are now want to discover the agents that kill the pathogen. That is some chemical agent, they want to discover some chemical agents that treat against the pathogen. Why? Why they want to try that? Try to discover that materials? Because the immune system sometimes fail sometimes fail to attack on the particular pathogen or action improperly. So inappropriately, right word, inappropriately. So disease may occur. So if the disease occur, then how will you tackle that? You need some drug, you need some chemical substance from the outside that can cure the symptoms. Yes or no? So scientists now try to discover some materials or some chemicals that can kill the pathogen. Okay? And that agents are called chemotherapy. These agents are called chemotherapy. Chemotherapy is a broad term. মানে আমরা আমাদের মাথায় একটা কি ঢুকে গেছে না, কেমোথেরাপি মানে আমরা ক্যান্সারের সাথে কানেক্ট করি, ক্যান্সার ইকুয়ালস টু কেমোথেরাপি। Yes, that's also a chemotherapy because that in cancer treatment also use some drug that kill the cancer cell. So any agents that kill the pathogens or other cells, the overall broad term is called chemotherapy. Clear? So chemotherapy means chemical substance that kill pathogen and that works against the pathogen. And this chemical substance can be of two types. Based on how does the chemical substance we are obtained. We can obtain the chemical substance in laboratory method. And second option, some chemical substance can also produced by now is it
Yes, sir.
okay? Some chemical substance can also produced by other bacteria and fungi. Some substance can be produced by bacteria or fungi to kill other microbes. So those chemical agents that kill the pathogen can be obtained from two methods or two ways. And based on that, one of them, this chemical substance can made laboratory in the synthetic means by simple chemical reaction by combining the molecules. Clear? Laboratory mode. And the second one, this type of substance can be produced by some bacteria and fungi also that neutralize or that kill other pathogens, other bacteria. So first, give an example. First name laboratory made compounds are called chemotherapeutic agents. They are called chemotherapeutic agents. And the second mode that produced by the bacteria fungi to kill other microbes, antibiotics. Antibiotics. Previously we stated about that antibiotics and the antibody, two confusing terms. We are interchange every time, but they are two different meaning. Antibody produced by our own WBC to kill or neutralize the pathogens, incoming pathogens, and the antibiotics that is produced by bacteria or fungi. That chemical substance we are isolating to make a drug to treat the disease. Clear? Two concepts are clear, antibiotic and antibody. Okay. So chemical substance can be of two types. One is laboratory made, other is produced by the bacteria fungi. The laboratory made compounds are called chemotherapeutic agents, and the substance that produced by the bacteria fungi to kill other microbes, they are called antibiotics. Chemotherapeutic agents, also called synthetic drug, because it's totally based on the chemical reaction. This chemotherapeutic agent was discovered by Paul Ehrlich. Which scientist? Paul Ehrlich. Paul Ehrlich first discovered a good chemotherapeutic agent and he named that magic bullet. Paul Ehrlich isolate first chemotherapeutic agent. So chemotherapeutic agent that discovered by the Paul Ehrlich, it is produced by the bacteria or fungi or it is produced from the chemical substance. Chemical substance, clearly. Mind should be clear, overall knowledge. Paul Ehrlich isolate the first chemotherapeutic agent known as salvarsan. Salvarsan. Salvarsan actually arsenic based compound. Which base? Arsenic base. Yes. So clearly it is a chemotherapeutic drug. It is made from the laboratory. Okay. Arsenic based drug that works against syphilis. Syphilis is a potent STD, sexually transmitted disease. This agent works good against the syphilis, bacterial disease, Treponema pallidum, syphilis causing agent. ISTD, one of the potent STD, syphilis, and affect both male and female genital tract. So salvarsan, he discovered the salvarsan compound, arsenic-based compound that works good against the uh, syphilis. And after that discovery, he used the term, used the salvarsan as term magic bullet. Magic bullet. Why? Why he given the term magic bullet about the salvarsan? Because it works magically. Salvarsan has it has two roles. Number one, it kills the pathogen specifically. Specifically pathogen, target way killing, number one. And it has also the less toxicity. So when you enter, when it given to the host show less toxic effect. Salvarsan has less toxic effect, and second, it works specifically against the syphilis causing organism. That's why he used the term magic bullet. Question answer. What is magic bullet and why Paul Ehrlich used the term magic bullet against the Salvarsan compound? Number one, it specifically works or targeted work against syphilis causing organism, bacteria. And number two, it has less toxicity. Every drug has toxicity, but salvarsan has less toxicity. Into the post. Clear? Who discovered magic bullet? What is the magic bullet component? Why it is called so? Various way question can become. Magic bullet is the antibiotic or chemotherapeutic agent. It is the chemotherapeutic agent, and give reason for that because it is the arsenic based compound produced by the Paul Ehrlich in the laboratory. That's why it is chemotherapeutic agent. As simple as that. Question can be various way can become, but the concept should be clear. Clear magic bullet, Salverson, Paul Ehrlich contribution.
Yes,
Online
sir.
student. Okay. Next, another substance. Second hand. Right hand. Some substance can produced by the bacteria that is not produced in the laboratory, produced by the microbes, produced by the living agents, biological agents that kill another biological organism. That's why it's called antibiotics. Just break down the words anti and biotics. Biotics means it kill the other biotic organism, living organism, microbes. That's why anti word is coming in the prefix antibiotics. Okay. Antibiotics first discovered by Alexander Fleming. First antibiotic is penicillin and discovered by Alexander Fleming. From the mold. Mold is the type of fungi. Penicillium notatum. Nowadays, it is termed as Penicillium chrysogenum.
Sir, could you please spell the last word?
Penicillium notatum. Full name, I write it there, P notatum. P means penicillium. Penicillium notatum. And nowadays, the notatum species actually called Penicillium chrysogenum. Chrysogenum. So better to say now Penicillium chrysogenum. The species name has been changed from notatum to chrysogenum. Better applied name Penicillium chrysogenum. And he discovered the first antibiotic, penicillin. Alexander Fleming. Do you know the story? Huh? He actually accidentally discovered the antibiotic, this concept. This concept is come accidentally. He left for 2 weeks. He is in the holiday mode for the 2 weeks, and when he returned, he observed that the staphylococcus, some staphylococcus plate, agar plate, the colony is there, but when the mold is grown, the outside, the periphery area is clear. That means no zone of no sign of growth. So from that view, he actually think that Penicillium notatum, that means the mold which is grown in the agar plate, produce some juice and that kill the growing microbes, Staphylococcus plate, Staphylococcus microbes. Actually, he forget to discard those plate. Discard. Actually, that okay, উনি actually discard করতেই ভুলে গেছিল যাওয়ার সময়। তো উনি ব্যাক করলো যখন তখন দেখছি যে প্লেট পড়ে আছে and প্লেটটাকে যখন ভালো করে চেক করছে, that time he saw that mold growing area no bacterial zone. So accidentally he discovered. তো বলে না যে anything are happen for the reason. So Penicillium notatum, penicillin production, discovery actually accidentally for discovered accidentally by the penicillin. আলেকজান্ডার ফ্লেমিং। মানে ওটা অ্যাকচুয়ালি ডিসকার্ড করে দেওয়া উচিত ছিল।
যদিও কিন্তু নরমাল যদি কেউ তো ফেলে
দ্যাটস দ্য অ্যাকচুয়াল রুল। তোমাকে কোন জায়গায় তোমাকে লিভ নিতে গেলে, উনি এক প্রফেসর ছিল, ব্যাকটেরিওলজি প্রফেসর ছিল হচ্ছে তোমার লন্ডনের সাম হসপিটাল নেম আই ফরগেট, সেন্ট মেরি হসপিটাল আই থিংক, অ্যাসিস্ট্যান্ট প্রফেসর ইন ব্যাকটেরিওলজি, and works do not against any penicillin producing organism. He works against the virus that time. Okay, influenza virus. And then the staphylococcus plates are there because his previous experiments, he forget to discard that plate. And when he got back to from the holiday and he observed that some plates are there and observe carefully, that time is very clear. That time is very clear, critical. Should you throw the plate out or just observing that? And that's the criteria that create the difference between the ordinary people and the extraordinary people. And the scientists always extraordinary category. That's create the difference. Ordinary people ki korto, throw, trash korto, dump korto, just me. But Alexander Fleming look carefully and observe the concept of antibiotic. Clear? So by that way, medical microbiology and immunology, two important subjects in our regular life nowadays, are established. Today's class done about this is class totally about the medical microbiology and immunology field discovery establishment. Next class we discussed about soil microbiology development as a subject. Soil microbiology. So there are also some scientists' contribution to establish soil microbiology, another field of microbiology. Soil microbiology and biology. And that will be the last class for that unit. Unit will be covered up. So next week.
Sir, I have a question regarding books. Sir, today I saw the Prescott book. Sir, ওর মধ্যে মানে 4 ইয়ারের সিলেবাসের মতন ছিল, মানে অনেক কিছু আছে, একটু কমপ্লেক্স ওয়েতে আছে। আর টেক্সট বুক অফ মাইক্রোবায়োলজি যে বইটা আছে ওর মধ্যে একটু সিম্পলিফাই করে দেওয়া আছে।
টেক্সট বুক অফ মাইক্রোবায়োলজি মিন্স আরপি সিং।
হ্যাঁ,
আরপি সিং copied book from Prescott, from Brock, from Black, just like my notes, copied from different book, adapted book. So RP Singh also a copied book. Huh?
So which one I should buy?
Prescott, clearly.
So তো প্রেসকটে দেখলাম কি ওর মধ্যে না মানে টপিকস হেডিং যে ইন্ডেক্সটা দেখলাম, হিস্ট্রি অফ মাইক্রোবায়োলজি এই সব বলে নেই।
সেটা আমাকে জেনে নেবে তো কোন পার্টটা পড়তে হবে। তুমি পুরো বই দেখলে তো ঘাবড়ে যাবে, ইউ উইল বি প্যানিক অ্যাটাক। কারণ এই পুরো বইটা এরকম একটা সাইজের, মোর দ্যান 2000 পেজেস আই থিঙ্ক। তো কোন পার্টটা পড়বে, কোন রিগার্ডিং, কোন ইউনিটের, দ্যাটস ওয়াই তো তোমার একটা গ্রুপ করা রয়েছে সেপারেটলি। আস্ক কোশ্চেন, পোস্ট দ্য কোয়ারি।
সো স্যার গ্রুপ ইউনিট 1 আর 2 এর জন্য প্রেসকট নেব?
তুমি সমস্ত কিছুই পড়তে পারো ফ্রম দ্য প্রেসকট।
আচ্ছা,
সেমিস্টার 1 এর জন্য প্রেসকট, সেমিস্টার 2 এর জন্য প্রেসকট, সেমিস্টার 3 থেকে চেঞ্জ হবে।
আচ্ছা ঠিক আছে,
ঠিক আছে।
থ্যাংক ইউ।
সেমিস্টার 2 এও যা সিলেবাস আছে প্রেসকট মোর দ্যান এনাফ, কমপ্লিট হয়ে যাবে।
And sir, one semester will be for 6 months, na? At the 6th month, the exam will take place.
After the 6 month, exam is take place, final exam. And before that,
মানে first semester এর জানুয়ারিতে হবে?
জানুয়ারি ফেব্রুয়ারি। প্রেসকট পুরো বই লাগবে না তোমাদের, আমি আবারও বলছি, প্রেসকট তোমাদের পুরো বই লাগবে না, বাট প্রেসকটটা কেনানো এটাই তোমার পুরো সেমিস্টার, টোটাল 4 সেমিস্টার মানে 8 সেমিস্টার তোমাকে সাপোর্ট করে যাবে। নট অনলি বিএসসি, অলসো ইন দ্য এমএসসি অলসো। ঠিক আছে, এমএসসি তো তোমাদের প্রেসকট বই লাগবে, কোনো না কোনো কাজে কোনো ভাবে একটা লাগবে, কানেকশন থাকবে প্রেসকটের সাথে, দ্যাটস ওয়াই প্রেসকট তোমাকে লাগবে। আর রেফারেন্স বুকে আমি ব্লগ বলেছি, চাইলে কিনবে, না হলে প্রিন্ট আউট ভার্সন ই বুক রাখবে। ঠিক আছে,
রেফারেন্স বই আবার একটু বলবেন?
ব্লগ মাইক্রোবায়োলজি, ব্লগ মাইক্রোবায়োলজি।
থ্যাংক ইউ স্যার।
আরপি সিং নিও না, আরপি সিং ইজ নোটবুক টাইপ অফ থিং, মানে আমি যে নোটগুলো নিচ্ছি, আমার যে নোটটা পড়ছো তুমি, লাইক দ্যাট আরপি সিং ইজ দ্য নোটবুক। তা আমি যেমন ডিফারেন্ট বুক থেকে কালেকশন করে নোটটা বানাই, আমি তো আর নিজে বানাচ্ছি না রে বাবা, আমি তো আর আকাশ থেকে কেউ দেয়নি আমাকে, ভগবান বলে গেছে যে এইটা তুমি লিখে যাও। আরে আমি তো নিচ্ছি কোনো না কোনো বুক থেকে। ইয়েস অর নো, একটা নোট কি করে প্রিপেয়ার করা হয়, আমি ফ্র্যাঙ্কলি বলে দিচ্ছি তোমায়, নোট প্রিপেয়ার করা হয় ডিফারেন্ট বুককে কম্পাইল করে। ঠিক আছে, ডিফারেন্ট বুক থেকে ডিফারেন্ট জিনিস নেবো, কারণ একটা বুক ইজ নট সাফিসিয়েন্ট, প্রেসকট ইজ নট সাফিসিয়েন্ট, বাট তোমাদের জন্য সাফিসিয়েন্ট, তোমাদের জন্য পড়ার জন্য নাও, সেমিস্টার 1 পড়ার জন্য সাফিসিয়েন্ট, বাট স্টিল কোশ্চেন লিখতে গেলে আরো কম্প্যাক্ট নলেজ দরকার হয়, দ্যাটস ওয়াই তোমার ডিফারেন্ট বুকস আছে। এবার তোমার ক্ষেত্রে তোমাকে যদি আমি বলি ব্রক পড়ো, প্রেসকট পড়ো, ব্ল্যাক পড়ো, টরটরা পড়ো, 4-5টা বই বলে দিই, তুমি কি পড়তে পারবে একসাথে, সামলে উঠতে পারবে? তুমি কোনোদিন পারবে? পসিবল? ইজ ইট পসিবল নাও জাস্ট ফর এ বিগিনার? নট পসিবল। দ্যাটস ওয়াই একটা সিঙ্গেল বুক, সিঙ্গেল অথার বুক রিড করো, যা নলেজ আসবে, তারপর তো আমার নোটস থাকছে, কনসলিডেট নলেজ, দুটো মিলিয়ে তুমি আনসার আরামসে করে দিতে পারো যে কোনো কিছু।
ওকে স্যার, থ্যাংক ইউ।
ক্লিয়ার? ওকে।